describe the anatomy of the nasal cavity. You can show me that there are bumps on the sidewall. It's called conchae, where the bones make these bumps. And then there are these nasal meatuses, these deep grooves that go up underneath the bumps. And it's coated with a well-vascularized mucous membrane. That mucous membrane. Remember, the whole idea was to create increased surface area of the mucous membrane. And the bumps and the grooves create turbulence in there. Does this sound familiar? The turbulence increases the collisions of air molecules with the moist, sticky mucous membrane that's also warm. And you're talking about how the nasal cavity would warm, cleanse, and humidify the air, right? This is the physiology of the nasal cavity. Let's see. We probably do it. I mean, we do something about that mucous membrane. What type of epithelium is found lining the luminal surface of the trachea? Ciliate, pseudostratified columnar epithelium. You're going to know it for the rest of your life?
What if you put pseudostratified ciliate columnar?
Oh, put the words in whatever order you want. I always figure that they have to do with what is your native language. Whether the adjectives go before or after the noun. Right? You know what I'm saying? So I don't think it matters in this instance what order you put those words in, but I definitely want you to see ciliated in there.
Yeah, I'm sure.
So, in order we do. Okay, so before our break, we were saying that respiration has 4 smaller parts to it, and we're going to go cover pictures that fit into this framework. So I'm about to take you to a picture that talks about inspiration and expiration, the 2 sides of the pulmonary ventilation column. The easy way to refer to that, of course, is just breathing. That's what it is, right? It's breathing, pulmonary ventilation. And I would ask you to meet me if you would on figure 16, I guess it is, figure 16. I'm thinking figure 16 is the single most complicated way to explain how we breathe in and breathe out. So let me see if I can simplify it. If I step away from the picture momentarily. Look at this. Let's say I have a balloon and it's partially inflated and it's tied off so the air that is in there can't escape. Does everybody recognize that if I were to squeeze the balloon so that it now takes up less space in the universe, if I squeeze the balloon, decreasing the volume of the balloon, meaning it's now fitting into a smaller space, if I squeeze the balloon, does everybody agree that the air that's in the balloon is now under greater pressure? Everybody see what I mean? Closed balloon, if you make it fit into a smaller space, then the air in there is under greater pressure. So there's this inverse relationship between volume, the amount of space it takes up, and pressure. If I decrease the volume, what happens to the pressure? Pressure goes up. Because I'm really interested at first in you seeing the opposite of that, but that's harder to visualize or conceptualize. Think about the same balloon. And then, while I don't think this is really possible, think about if I could do this to the balloon. If I could grab it and make it take up more space. If I could grab it and somehow make it larger, that's really hard to imagine happening. But if I could somehow do the opposite of squeezing, if I could make it larger so that it now takes up more space in the universe, what happens to the pressure on the air in the room? Pressure would go down. Everybody get it? Increased volume, decreased pressure. Decreased volume, increased pressure. See, there's that inverse relationship. Volume goes up, pressure goes down. Volume goes down, pressure goes up. Okay. This is all we do to breathe. Watch. You know, you already probably have an idea in your mind about how you breathe in. Look, you take the lungs and you stretch them open, and which draws air into the lungs. Right? That is as simple as you can make it. You already inherently understand that if I pull the lungs open, air is going to get drawn into them. But our book wants to put a step in between there where they're asking us to understand that relationship between volume and pressure. Watch. Why is it that when I pull the lungs open, that air gets drawn in? Why is that? Here's why. Because when I pull the lungs open, just like the balloon, they're going to take up more space. So the volume increases, and now the pressure on the air in there goes down. Everybody good? If I pull the lungs open, the air that's in the lungs becomes less pressurized. Now it becomes, let's say, 759 millimeters of mercury. Out here it's 760, but because we just stretch the lungs open, the pressure in there drops ever so slightly. And And now what we've got, everybody, is a good old-fashioned gradient. 760 out here, 759 in there, so the air simply flows down its pressure gradient from higher pressure out here to lower pressure inside the lungs. Does that make sense? They're asking us to add this step of understanding in between, but it's just as simple as saying this: if you pull the lungs open, air gets drawn in. You understand that. They just want you to understand that the reason why it gets drawn in is because we've dropped the pressure on the air in the lungs, and now air flows down its pressure gradient. If you're okay with that, then you understand already the top band across this figure 22.16, which is just talking about breathing in, or inspiration. Watch what they said. Here's how you breathe in, everybody. You contract your inspiratory muscles, the muscles for breathing in. Now, which muscles are those? Well, it includes your external intercostal muscles, between the ribs, the external ones now. When you contract those, here's what happens. The rib cage gets spread further from left to right, and the sternum gets pushed forward from your spine. So the rib cage, the thoracic cage, is expanding in volume from left to right and from front to back. Everybody good? And also what these external intercostal muscles do is this. They elevate every single rib up to the rib above it. They pull all the ribs up so you get this. The whole rib cage elevates. So the external intercostals raise your rib cage, spread it from left to right and front to back, and at the same time you're contracting those, you also contract that dome-shaped diaphragm in there. And when you contract the dome-shaped diaphragm, it flattens out. So remember that the lungs are stuck to the inside of the chest wall, and they're stuck to that diaphragm. So when the diaphragm drops and the rib cage elevates, you're stretching the lungs from top to bottom. And you're stretching the lungs because you're stretching the thoracic cavity from left to right, you're stretching the lungs from left to right. Because you're stretching the thoracic cavity from A to P, if you will, in the anterior to posterior dimension, then the lungs are getting stretched. Remember, the lungs have no choice but to expand with these structures that we see here. So we've got these two sets I want you to know of inspiratory muscles, the diaphragm and the external intercostals. What do they do when they contract? They expand the thoracic cavity's volume. And if you understand that the lungs are suctioned to the thoracic cavity in the diaphragm, then you understand that increasing the thoracic cavity volume also increases the lung volume. In other words, the lungs are getting pulled open. And then this drops the pressure in the lungs, and you don't have to drop it much. You just have to drop it by maybe, say, 1 millimeter of mercury is all. But that's enough to set up a pressure gradient. 760 out here versus 759 on the inside of the lungs, and now the air just flows down the pressure gradient. And it flows in there until we equalize that pressure, until it's 760 and 760. See, they make it very complicated, but it's really a very simple idea. Stretch lungs over, air strong. That's how you breathe in. But it did let us introduce the inspiratory muscles and see how those move the rib cage and expand the lung volume. That's the story of inspiration. And now we'll talk about expiration. Remember me saying this earlier, and I guess I'm going to say it a lot of times. If you're breathing just restfully and calmly, you don't need to contract any muscles to breathe out. You breathe out passively, simply as a result of the recoil of the elastic structures of the thoracic cavity and the respiratory tract. Watch. In order to breathe out now, expiration, we'll blow up that expiration process. It's an even easier story to tell because there's no new muscles to introduce. In order to breathe out, all you have to do is stop contracting those inspiratory muscles that help us breathe in. As soon as you stop contracting them, as soon as you relax them, here's what happens. Everything recoils due to the elasticity. So the rib cage comes back in from left to right, the sternum comes back towards the vertebra, the rib cage descends again, the diaphragm resumes its resting dome shape, and now the lungs that are stuck in there are getting squeezed. So just like that balloon I started talking about, where I started off by saying what would happen if I squeezed it so it took up less space? Pressure there would go up. Same thing's happening to the lungs now. The recoil of all these structures, the lungs are getting squeezed, like the balloon, and that means that the air in there is becoming increasingly pressurized. The thoracic cavity volume decreases, which means the lung volume is decreasing, because they're stuck together. And that pressurizes the air in the lungs, and you don't have to raise it very much, that pressure, just by 1 millimeter of mercury worth of pressure. But now, you've set up a pressure gradient. Now it's like it's 761 inside of your lungs and 760 out here. So the air flows down its pressure gradient from 761 in the lungs to 760 out here in the external environment. So the air flows out of the lungs down its pressure gradient until we reach the lung. It's really a very easy story. They make it, I think, unnecessarily complicated
But they do want us to understand that relationship. If we decrease the volume, we increase the pressure, the less you can set up a gradient. Or if we increase the volume, we set up a gradient by dropping the pressure in there, and that draws the gradient. So I want you to see that this picture, figure 16, would fit into our framework under the topic of pulmonary ventilation. So we just talked about these two processes, right? Inspiration and expiration. But there's more to talk about here under pulmonary ventilation, because that breathing in and breathing out that we just talked about is controlled by neural signals coming from your brain. And as it turns out, someone has done a good job of mapping out what brain regions are controlling the breathing and the actual neural pathways from those brain centers that control inspiratory muscles, turning them on and off. So now I'm going to ask for you to jump with me. I know, you don't surround the chapter. I'm going to ask you to jump with me to figure 26. So we need to talk about the neural control of breathing and breathing out. Hey, do you recognize this picture at the top of figure 12.26? Does everybody recognize that that's a mid-sagittal view of the brain? And maybe I better stand over here. This is a picture of the right half of the brain, but you're viewing it from the left. So it's as though the brain were cut like this, but we're taking the whole left side and discarded it. And then we're viewing from the left, we're viewing the right side. So it's a mid-sagittal view. Do you recognize this to be the cerebellum back here? I always try to teach students to use that as your landmark for knowing posterior. So it's a side view from the left, like you're looking at my head here. This is posterior. Your eyeballs would be here looking that way. It's good? Everybody got it? Okay. So remember that the brain stem, then, everybody, is where the spinal cord comes up and then becomes the brain stem, where it becomes the medulla oblongata and that egg-shaped pons. And above there, we've got a little area called the midbrain. Well, that doesn't fit into our story. But they blew up the picture of the brain stem. Remember the egg-shaped pons here? And then the medulla oblongata, which is practically like the top of the spinal cord. The pons and the medulla are part of this story of the neural control of breathing. The brain centers that control breathing are located in your brain stem, almost like in the back of your throat. By the way, the adjective form of pons is pontine. Also seems unnecessarily complicated, but P-O-N-T-I-N-E. So we're about to introduce this group of neurons here called the pontine respiratory centers. That's just telling you that they're located in the pons. In the medulla, we've got medullary respiratory centers. Medullary, because they're in the medulla is all, right? And there's one in the front, so we're going to call this one the ventral respiratory group, and there's one in the back, so we're going to call this the dorsal respiratory group. And I'm hoping you realize that these orange spots are really this. Clusters of neuron cell bodies that we would have identified in our AMP1 class as being nuclei. A nucleus is a group of neuron cell bodies in the central nervous system. There are lots of named nuclei. These are respiratory nuclei that control respirations or breathing. They're brain centers, they're brain regions, they're clusters of neuron cell bodies. And the main player of the three that are introduced here, the pontine respiratory center, is actually multiple ones. They made the big group, we call it centers. That's better than if they had given us all the separate ones. It's easier. There's one in the pons that we need to know about, and we need to know about these two in the medulla, one in the front, one in the back, ventral and dorsal. The main player in our story is this ventral respiratory group, the VRG in the medulla, the ventral respiratory group. Here, there are neurons. These are the cell bodies now, even though their axons may travel far away. The neuron cell bodies reside here. These neurons here have been shown to do this, to spontaneously depolarize. That means they become electrically excited, generate action potentials. We used that phrase, spontaneous depolarization, before. You know where? When we talked about the SA node in the heart's intrinsic conduction system. You remember that? And they said, oh, look, the SA node spontaneously depolarizes 75 times a minute, setting your heart rate. Everybody okay? So you know to spontaneous depolarization. Same idea with these neurons here. They act just like the SA node, except they depolarize, these neurons do, about 12 to 20 times per minute. And every time they become electrically excited, they launch out a barrage of action potentials And the neural pathways through which these action potentials travel have been mapped out. Those neural pathways lead us to the external intercostal muscles and the diaphragm. So these neurons become electrically excited, they launch a barrage of action potentials telling these muscles to contract. The muscles contract and you do this. You breathe in. Inspiration. Those are the inspiratory muscles. We just talked about what would happen when you contract them. On the last picture, you expand the thoracic cavity, drop the volume, drop the pressure in there, and then the air moves in, down, pressure in. This is how you breathe in. 12 to 20 times per minute. This respiratory center launches action potentials asking you to breathe in. Now, if you're breathing restfully, remember what it takes to breathe out? You just have to stop doing this. Tell me, breathe in.
Is that just when you're breathing and not thinking about it?
Yep,
Like,
that's just when you're breathing and not
If you're
thinking.
exercising and you know you need to breathe
We're
more.
going to get to that.
Oh,
So if we're just doing normal restful breathing, then these ventral respiratory group neurons are depolarized somewhere between 12 and 20 times a minute spontaneously without any outside input, and they cause every inspiration. Now, how do we get an expiration? All you do is stop inspiring. Remember, there were no new muscles involved. You just have to stop contracting your inspiratory muscles, and then recoil takes care of it all for us. So here's what happens. You ready? These VRG neurons become electrically excited, start launching out action potentials, and as soon as they do start launching out action potentials, they get turned off. They immediately get turned off. How do they get turned off? The pontine respiratory centers inhibit the ventral respiratory group. And it seems to me that on an old version of this picture, they had a valuable negative sign, like a nut, like to show that it's inhibitory, on this little black arrow from the pons down to the ventral respiratory group to try to show you that it's inhibitory. Every time the ventral respiratory group becomes electrically excited, starting in inspiration, we're going to immediately stop the inspiration. And then recoil happens, and you breathe out. How do we stop the barrage of action potentials coming out of here? The pontine respiratory centers turn off the VRG every time it turns on. And that leads us to the next expiration. Does that make sense? And then, because these guys are on a 12 to 20 times per minute cycle, they become electrically excited again. Some action potentials get sent out, so you start to breathe in. The pons turns it off. And as soon as you turn it off, everything recoils and breathe out. And it keeps happening like this. Watch. Look what it says over here. The ventral respiratory group contains rhythm-generating neurons whose output drives, see where it says their output drives respiration? I sent them an email. I say, could you just be a little bit more specific there and say that their output drives inspiration? It does. When these neurons send out electrical signals, you get an inspiration. The expiration only comes up when you turn these things off. How do you turn them off? The pons does that. But they didn't say that clearly enough here. You're going to have to dig it out of the paragraph. So I dug it out for you because I didn't get it on here. I wish they had. It says that the pontine respiratory centers help to smooth your respiratory pattern. They do. Have you noticed you don't breathe like this? There's not like a sharp transition in between inspiration and expiration. Like if you saw it on a graph, it's not jagged like the design on Charlie Brown's shirt. Charlie Brown, everybody, yellow shirt, black zigzag across. If you saw it on a graph, it's not like Charlie Brown's shirt. It's a wave. Every inspiration gradually ends and becomes an expiration. And then the expiration gradually leads us to the next inspiration. It's a smooth pattern. The pons is turning off the ventral respiratory group in a very gradual way. So that we transition nicely from inspiration to expiration. Yeah, that's great.
So, like, if you're just kind of resting and then you realize, like, oh, I need to take a deeper breath, and so you breathe in more deeply and
Yeah.
you exhale, are we overriding that whole system?
Yes, we're still using this system. We're just sending more signals to this system to ask it to breathe more deeply or more frequently. And we're going to try to get to that here shortly. Check this out. So now we've introduced this second player. What's its main role? To turn off each inspiration, which really begins the expiration, just because turning off inspiration, you're going to get that. That's really practically the whole story, except there's this. Now we're getting to the questions that came up, which is this. Well, what if you have other circumstances going on? What if, let's say, you're exercising your muscles vigorously? Or what if your body has detected that there's not enough oxygen in your tissues or not enough oxygen in your blood or something? look, all your body parts everywhere are bombarding your brain with sensory signals about their conditions or in all those different body parts. And if they're sending sensory signals that say we need more breathing, then we're going to increase the rate of the ventral respiratory group's depolarization. Maybe instead of 12 to 20 times a minute, maybe you're now going to breathe, I don't know what, 16 to 26 times a minute. See what I mean? Or if your tissues out here are all detecting that they're not using oxygen very rapidly, they could send inhibitory signals in here that are saying, we got plenty of oxygen, you could slow it down a little. And then the VRG would reduce the number of spontaneous depolarizations per minute. Everybody see what I'm saying? Well, that's where I have to introduce you to this third player. The dorsal respiratory group, towards the back, dorsal. The dorsal respiratory group has been shown to coordinate multiple sensory inputs. It seems to listen to all your body parts and determine, should we increase or decrease our respiratory rate and the depth of our breathing? And then based on a sensory picture that it gets, it's going to influence that ventral respiratory group. See that little tiny short arrow? That little tiny short arrow used to have a plus and a minus associated with it in the past picture. And that was helpful. There's really not room to wedge it in there. But I want you to know that based on sensory inputs, the dorsal respiratory group can now increase or decrease the activity of the ventral respiratory group so that we're going to breathe either more or less. And I think that starts then to get to the questions that people have, which is, well, what about under certain circumstances when you're not just breathing restfully? So what sorts of body parts are sending in these sensory inputs that are coordinated by the DRG? We're going to get to that on the next picture. But wait, before we do, look at the description they give of the dorsal respiratory group. I couldn't have done it better myself. Look. It integrates peripheral from all over the body, peripheral sensory inputs, and then modifies the rhythms that are generated by the ventral respiratory group. Isn't that exactly what I was trying to describe? They did it better. They did it more concisely and very, very clearly. It's listening to sensory inputs coming from all over the body. Okay, but where are those sensory inputs coming from? That's figure 27. On figure 27, they made those few orange spots, those respiratory nuclei we just identified in the last picture. There's a lot more nuclei than that. So they just made a big area of the brain stem here, as opposed to showing a separate named little nuclei. They just said there's a lot of sensory inputs that come to the brain stem. But they're trying to show you where these sensory inputs come from, and I want you to see this. Check it out. Previously unintroduced to you, I want you to now see that in the aortic arch, on the walls, literally, of this blood vessel, there are special cell populations. And also present at the bifurcation of your carotid artery into the internal and external carotids. We can talk about those blood vessels in here, but I'm sure you had to in lab. Right? Know those? So at the bifurcation of the carotid, we have what we call carotid bodies, and in the aortic arch, we have what we call aortic bodies. They're special cell populations that are doing this. They're detecting certain chemicals in the blood, so we call them chemoreceptors. There are chemoreceptors there, and they're detecting if there's not enough oxygen in the blood, or if there's too much CO2 in the blood, which these two things are very much saying the same thing, I hope you would realize. Either we don't have enough oxygen or we have too much CO2. Those things are always occurring together. But somebody was able to show separately that there are separate populations of cells detecting these two things separately. So they're putting them both up there, even though they occur at the same time. And then look at this. Look at what else these chemoreceptors can detect. When there's too much of that H plus in there. Do you remember us introducing this H plus earlier in the semester and me saying, that's the nasty acid, everybody. That's acid. That's the hydrogen atom from the periodic table that has lost its electron. Now we call it the hydrogen ion, or we call it a proton. And I want you to see that as acidity. I'm going to try to convince you coming up that whenever there's more CO2 in the blood becomes more acidic. And it's a little tricky that they point with the H's going up. It is that the H's are going up, but that always means that your pH number on the pH scale is going down to become more acidic. So whenever there's more CO2, the pH goes down, or the blood becomes more acidic. You get too many free H pluses, I'm saying with an F. There's too many of these free-floating H pluses in the blood. So these three conditions really are saying the same thing 3 different ways. And what they're saying is we need to breathe more. There's not enough oxygen, there's too much CO2 or too much acid. They occur all at the same time, but they've just been able to show separately that these things are individually detected. So we're called these peripheral chemoreceptors, and look, they're stimulatory to the respiratory centers, which means that they get you to breathe more frequently and possibly deeper. Somebody asked about breathing more deeply. So you're going to get more frequent and deeper breathing, and you can correct these things in a few minutes. This is going on unconsciously all the time in your body. You don't even think about it. You don't even realize that you breathe 3 more times last minute to correct for a little bit of acidity in your bloodstream. You do it all the time, unconsciously. Okay, now wait. Somebody's also been able to show that these same sorts of chemoreceptors are located in various spaces, various places in the brain. So they're going to call those central chemoreceptors. No one's found one yet that detects decreased oxygen. So they just listed, there's still the same idea, that there are special chemoreceptors. If we have too much CO2 or too much acidity, we'll want to breathe more, so we stimulate the respiratory centers. How about this? If you were to start using your muscles vigorously, I want you to see that that would lead to these chemical changes in the blood. Because if you start using your muscles vigorously, the muscles are going to pull oxygen from the blood, use the oxygen in the muscles, and the muscles are going to start spewing out CO2 into your blood. You're going to get these changes. And whenever you get CO2 in the blood, you're going to get acid in the blood. Plus, vigorous muscle activity is going to produce some lactic acid. You're going to get some of that in the blood. You're going to have a lot of this. Everybody see that increased muscle activity will lead to these changes? Well, before it even leads to these changes, as soon as you start using your muscles, there are special receptors in the tendons and in the bellies of your muscles that immediately send signals to your respiratory centers that say, start breathing more now, even before those blood changes occur. As soon as you start using your muscles, the brain is informed, we're going to need some more breathing. So these sensory receptors are sending in also stimulatory information. There are some sources of inhibitory information, like if you were to overstretch the lungs, then we don't want to overstretch them and damage them, so we'll inhibit our breathing if there's too much stretch perceived in the lung, or if there's irritants in the air that we're breathing, you might not want to continue to breathe in as deeply when there's poisonous or toxic air, so then we might be able to inhibit these respiratory centers. There's more. Somebody asked, I think, about conscious control. Does everybody recognize the gyri and sulci, the bumps and the grooves that give us that noodly looking appearance to the surface of the brain? You remember this, everybody? That those bumps and grooves are there to give us increased surface area so that we can get more of this, more crust on the outside of the brain. It's that crust that only goes a few millimeters thick that we call the cerebral cortex. The cerebral cortex is gray matter, meaning that it's just millions and billions and billions of neuron cell bodies, gray matter. And white matter is where we have myelinated axons, right? This is gray matter, covering all the bumps and the grooves. And those neurons that reside in that gray matter, right on the surface of your bumps and your grooves, that's where consciousness resides. That's where you do all of your conscious thinking and your decision making. It all occurs in the outer few millimeters of brain tissue out there. So the cerebral cortex can be used to consciously stimulate or inhibit these respiratory centers, increasing or decreasing the amount of breathing. Watch me do it. Ready? Watch me use my cerebral cortex. Look, I wouldn't say that I have overridden what's going on here. I just deliberately sent stimulatory signals that increased the output from my VRG. Everybody get it? Or I could also consciously inhibit this, right? I could go like this. And I could hold my breath. I can use consciousness, the deliberate thought, to inhibit these respiratory centers. Now wait. If you inhibit these respiratory centers consciously, you try to hold your breath. I want you to see that in a matter of just a few seconds, maybe a minute, you're going to start to have these blood changes. And then these sensory signals are going to override your attempt to inhibit this. Everybody see what I mean? You can only hold your breath so long, then you're going to breathe again. I say this because this is how the physiology is understood to occur, is that these signals here would take precedence and override your attempt to inhibit those respiratory centers. And I say that because I have often had somebody say, no, I'm telling you, when my 4-year-old daughter doesn't get her way, she will hold her breath till she passes out. I think this is a myth. I've never seen this happen. I've had students tell me, I'm telling you, you get a stubborn enough kid, they'll hold. I never saw it. I don't understand the physiology that way. I believe that they can try as they may to hold their breath as long as they want, but the blood changes are going to happen, and then these sensory signals are going to override their attempt to block breathing, and they will breathe. Everybody okay? Just wait, and let them, they'll start breathing again. You don't have to call my own. Also, does everybody remember the hypothalamus? I mean, we didn't really cover it adequately in our class, which is why I made you do a take-home quiz, right? Did I collect those? Give you a 10 on the take-home quiz? Take-home quiz on the endocrine system?
Yeah.
Did you get a 10? Did you get your 10? All right. Well, I'm hoping that you learned something about the hypothalamus there, and but it really didn't cover very much about how there's emotions here. And emotions can have stimulatory or inhibitory effects on these respiratory centers too. Oh, we spent too long on this page. We spent way too long. I just want you to see that there are lots of sensory inputs, and ultimately lots of sources, let's say, of sensory information coming from all over the body. And all of these sensory inputs ultimately are coordinated by that dorsal respiratory group, and then based on the sensory picture, it's going to increase or decrease the output from the VRV. See, these two pictures, 26 and 27, in my mind, these pictures are talking about breathing. We're still talking about pulmonary ventilation. We're just talking about the neural control of breathing in and out is all. And then I got one more picture under the topic of pulmonary ventilation, one more picture to show you. And that's figure 16, I'm thinking maybe. Let me see. No, 19. Figure 19. Oh, carotid body. So in the aorta there are aortic bodies, in the carotids there are carotid bodies. It's just a name given to the structure that somebody identified where these chemicals are being detected. Hey, check out figure 19. There's a little explanation that happened. Well, I guess that's all we ever do. But there's a little explanation required for us to understand what we're looking at here. Did you see anything like this in lab? Anybody? Did you do the respiratory physiology lab where you had to blow into a tube? Okay. All right, so some of you did that and some of you didn't. That exercise that you're supposed to do in lab is a really sort of elaborate way of using a diagnostic instrument called a spirometer. A spirometer measures your respirations. I'm hoping you see the spire part of the word. That is, of course, the root word in respiration, right? A spirometer measures a patient's respirations. Now, if you ever find yourself clinically having to do a spirometer test on a patient, the spirometer is going to be a little electronic device, maybe about the size of this audio recorder here, and it'll have a little tube with an exchangeable mouthpiece, so you can always use a sterile mouthpiece on there. But it just needs to be a little tiny electronic apparatus like this now. Of course, the one that you use in lab is like this antiquated mechanical thing, because they want it to look like a piece of lab equipment, really. That's a type of a spirometer called a kymograph. I don't know, maybe somebody said this word in the lab. It's just a different type. It's just a crude, mechanical sort of a type of a spirometer that you would use in electronic more clinically. Anyway, the printout that you get might be called a spirographic record. That's what it's being called on here. Or you might call it a spirogram. The printout, like a telegraph machine gives you a telegram. Anybody? A phonograph machine. The graph is always the machine, and the printout you get is always the graph. Anyway, I want you to see the respiratory pattern that you can appreciate using a spirometer machine, and I want you to learn the names of these particular respiratory volumes that are shown for us on here. Not the numbers so much, that's not really the important part, but the definitions of those things. So watch this. With the spirometer test, you can trace somebody's breathing. When they breathe in, you'll see the graph go up like this. When they breathe out, you see the graph go down. They breathe in, they breathe out, they breathe in, they breathe out. When they're breathing calmly, like this, like you should all be doing in your seats now. That amount of air that's being exchanged during normal restful breathing is called the tidal volume. It's often abbreviated TV, tidal volume. And I think it makes sense because it's sort of like the tides of the oceans, right? You know, the waves come in, the waves go out, the waves come in, the waves. There's a waveform going on here. You know, you breathe in, you breathe out, you breathe in, you breathe out, just like how the waves come in and out here. So it is tidal. That's pretty good. You don't have to remember that that's about a half of a liter. I never remember it until I'm looking at this thing. It seems to me that clinically the numbers are always provided for you. But I do want you to see that it's a very narrow band that you're really using when you're breathing calmly. Everybody see that? You're breathing in a very narrow piece of your total potential range of breathing that you can do. So of course, when you're breathing tidal in a tidal volume way, then you are capable of breathing in way more deeply than that, aren't you? Look, if you were to breathe all the way in as far as you could, and breathe in as much air as you possibly could over and above tidal volume, then all that extra air you could breathe in over and above tidal volume is very sensibly called your inspiratory reserve volume. I couldn't have thought of something better or more descriptive to call it myself. Look, the IRV is the inspiratory reserve. It's the amount of air you can breathe in over and above your tidal volume. Now, since that tidal volume breathing is occurring somewhere in the middle of your respiratory range, it also means you could breathe out a lot further than that too, if you wanted to, right? You could also force a lot more air out of your lungs. If you were to force all the extra air you possibly could out of your lungs, I always say below tidal volume down here, all that extra air you can push out, that's called the expiratory reserve volume, or ERV. So I want you to see that these names all make good sense. Maybe it'll feel a little bit less like memorization if the names make sense to you. Tidal volume's in the middle. We've got some inspiratory reserve above and some expiratory reserve below. If you were to add together the three of these, all the blues, the lightest blue, tidal volume, the medium blue, IRV, and the darkest blue, ERV, if you were to add all of those together, then you've got yourself the entire volume of voluntarily exchangeable air. And we give a name to that. See how the bracket provides the definition, everybody? That's the vital capacity, we abbreviate it VC. What's the vital capacity? The total volume of air you can voluntarily get in and out of your lungs. It's all the blues combined. Does everybody see that the vital capacity is the same thing as adding together the inspiratory reserve plus the tidal volume plus the expiratory reserve? Everybody see that you add the three blues together and you get vital capacity? Everybody get it? Because right below this on the same page, it's part of the same picture, they give you all these like alphabet soup formulas down here. And I don't want you to try to memorize these formulas. That's crazy to try to memorize those. Instead, I would rather if you just understood the definitions of these things, like what is the vital capacity? Oh, it's the entire range of voluntarily exchangeable air. I want you to know it from the picture and be able to see that that would mean that we're adding together the IRV plus the TB plus the ERV. And you got the vital capacity. Because if you try memorizing those letters, it's never going to work out for you. It's better to kind of understand it and see it and understand what the definitions are. In lab, the respiratory physiology experiment, which often doesn't work out that well. So some labs might not even do it. Dr. McGovern was just saying tonight that he thinks if your lab got canceled too many times already this semester, he may skip it entirely. But look, in the lab, this is how it's supposed to go. You've got this device with a tube and a sterile mouthpiece, right? Everybody gets their own mouthpiece. And you're supposed to do this. You're supposed to breathe all the way in and make what we might call a maximum inspiratory effort. So you've got every ounce of air you can possibly fit into your lungs, like this. And then you put the mouthpiece in. So you breathe all the way in, and you're here, and then you put the mouthpiece in, and then you're told to blow your air into the mouthpiece. You blow out your IRV, you blow out your TB, you blow out your ERV. And it's kind of fun in the lab because people want to outdo each other. And so people are trying to push every last ounce of air out of their lungs, and people are like turning red. It looks like somebody might soil their underwear. And it gets a little bit fun in the lab. You're trying to push every ounce of air in. And then the test ends here. So what you're really getting a measure of is from the top to the bottom. Right? We started the test after we made a maximum inspiratory effort. And now from here, we're going to measure how much air comes out when we make a maximum expiratory effort. So you're supposed to really be measuring this, the vital capacity in the lab. Everybody see how it's supposed to work? And then you may do some calculations from there and so on. But I want you to get the idea of what you were supposed to do. Now check this out. Let's say you push all the air you can possibly push out of your lungs. You pushed it all out. I want you to see your lungs are not empty. When you forced every possible ounce of air out of your lungs that you can push out, you're only here. When you've used your respiratory muscles to push air out, that's as far as you can go. It's here, and you still have a substantial amount of air in your lungs. In fact, it's about 20%. It's about 1/5 of the total capacity of your lungs. Look, 1,200, and if you add all this together, it would be like 6,000. So 1,200 over 6,000, it's 1/5, 20%. There's 20% of the air left over that you cannot voluntarily remove out of your lungs ever at any given point voluntarily. We call it the residual volume. Could you think of something better to call it? It's the leftover, it's the residue. It's what's left over, it's the RV, the residual volume. What is the residual volume? It's the air left in the lungs after you've made a maximal expiratory effort. After you've pushed all the air out, you can voluntarily push out. It's the air that's still left in the residual volume. Now, whenever I say this, that you can't ever push that air out of your lungs, I'm fearful you're going to think, wait a minute, does that mean that there's old air from 1969? Or is there like old? No, there's never old air. This amount of air is always being turned over. It's always in a different place. It's always being renewed. So there's not old stagnant air in there someplace. It's just that at any given instant, you just can't get all the air out. It's always different air. But I do want you to know that air can come out of your lungs if you got the wind knocked out of you, or if you're choking on a hot dog and somebody gives you the Heimlich over. They're going to hit you in the stomach hard enough to get some of this extra air out and hopefully use it to expel a hot dog out of your airway. Or, I always say, well, if you got run over by a truck, you could get the air out of there, but you can't get it out using your own respiratory muscles. Residual volume. I think it's important for you to think of that residual volume in this way too, which is, I'm going to bring up a picture we used earlier tonight. Remember that this is called external respiration, the exchange of gases between the blood and the alveolar air. I want you to realize that even if you blew all of your air out of your lungs and held your breath with as much air out as you can get, you held your breath at the bottom of your ERV, if you held your breath all the way out there, there's still plenty of air here. And there always has to be some air here because the blood's going to keep moving. And the blood has to have some air to accept oxygen from and some air to dump its CO2 into. This process must continue uninterrupted. This external respiration, we called it. And having residual volume means that there's always some air here. It helps to ensure that external respiration won't be interrupted. But I want you to also see that if we were to now add together this amount of air that you can't get out of your lungs voluntarily, if we were to add that to all the air you can voluntarily exchange, then we've got this, the total lung capacity, the TLC. So the total lung capacity is all the voluntarily exchangeable air and all the not voluntarily exchangeable air. Not voluntarily exchangeable, voluntarily exchangeable. Total lung capacity. Okay, so wait, what's the total lung capacity again? It's all the voluntarily exchangeable air, called vital capacity, plus the air you can't voluntarily exchange, called residual volume. It's the vital capacity plus the residual volume. That's what the total lung capacity is. Is that okay? See it on this picture instead of trying to memorize the alphabet soup on the bottom. And remember also that I crossed out these other two terms that I don't think we ever need, because then those make it more complicated. How about this? Could you use the terms total lung capacity, and residual volume, could you use those terms to provide a definition of vital capacity? What? Here's what I mean. If I asked you to define define the term vital capacity using these terms, total lung capacity and residual volume, how would you do it? You would say, oh, well, the vital capacity is just this. It's just the total lung capacity minus the residual volume. Everybody see it? Dare I tell you that that's algebra? What you just did? Because once I tell you that you just did algebra, you're like, oh, I can't do algebra. So I don't know. But you did understand when I said that you just have to do total lung capacity minus this residual volume, and you're left with this. Or vice versa, right? You could manipulate those things back and forth. Vital capacity plus residual volume equals total lung capacity. Okay, let's see. Well, I don't want to push my luck too far, but I do want to refer to this specific area right here that we defined as the expiratory reserve volume. Because I want you to see that in order to ever get that extra air out of your lungs, I'm not talking about this residual volume now, everybody. I'm just talking about this. Pushing the air out below your tidal volume, voluntarily doing that. That requires you to use muscles of expiration, and we have deliberately avoided saying their names up until now. Because I wanted to discuss with you tidal volume breathing earlier that requires no muscles of expiration. It just involves turning on inspiratory muscles, then turning them off. Turning them on and turning them off, but only inspiratory muscles, right? Now, to get expiratory reserve out, you have to make a forced expiration. You have to force extra air out, and that involves muscles of expiration that we have not yet named. So the muscles of expiration, I always try to call them the muscles of forced expiration to make sure that there's no confusion. The muscles of forced expiration are these. The internal intercostal and the abdominal muscles. So think about this with me. Internal intercostal muscles, everybody, do the opposite of the external intercostals. Here's what they do. They pull every rib down towards the one below it, and it makes your whole rib cage do this. It makes the rib cage come down from top to bottom. It makes the sternum come in, and it makes the rib cage come in from left to right. We're squishing the lungs further to drive more air out, right? You know, just like my balloon. Decreased volume is going to increase the pressure and it's going to drive air out. Good? Okay, how about the abdominal muscles then? Why are they involved? Well, when the diaphragm is relaxed, it's dome-shaped, right? But if we compress the intestines underneath the diaphragm, by contracting our abdominal muscles, if we compress the intestines want to go up and down. And some of the intestines are going to get pushed up to the already dome-shaped diaphragm and force it up even further. So you're going to be jamming your diaphragm up further into your lungs, squishing the lungs further from inferior to superior. We're trying to squeeze the lungs to drive more air out here. And you do that with abdominal muscles by squeezing your intestines up into the diaphragm. So abdominals and internal intercostals are the muscles of forced expiration. Again, for blowing out birthday candles, or playing a wind instrument, or singing or whistling, or yelling, or trying to project your voice like I'm doing now. Okay. Big warning. There's something counterintuitive here. Internal intercostals are not involved in inspiration. They're involved in expiration. The prefixes are crossed up. External intercostals are involved in inspiration. See the in the X prefixes? Internal intercostals are involved in expiration. It was the external intercostals that were involved in inspiration. So beware there. That's sort of counterintuitive. It would all be easier and there wouldn't be that confusing issue if the muscles were properly named superficial and deep, like they should be named, instead of external and internal. The book breaks its own rule. They told us in chapter 1 that we should describe something closer to the surface as being superficial and something further from the surface as being deep. But when they named these two layers of muscles for us, they call one external and one internal. And they should call them the superficial intercostals and the deep intercostals. Then we wouldn't have confusing prefixes that are the exact opposite of what their function is. Anyway, in and X, so be careful there. Okay, now I think that we're done covering the 4 pictures under the topic of pulmonary ventilation. And now we have 2 pictures left in the chapter to cover. One under the topic of circulation, but you're already pretty expert about this. So we really just have one new phrase to introduce there. And then the last picture that we usually don't get to tonight. So let's get to this last, this circulation picture, knowing that we'll probably get to the next one until next week. Check out with me figure. There it is. Figure 20. Check out figure 20. It's very similar, I guess, to figure 1, now that you add the figure 1 in there. But it's something I think we all understood well for our last test. I think we all understood fairly well where in the pulmonary circuit you should expect to find blue, indicating low oxygen levels, but remember, of course, that the blood would be red. And where in the pulmonary circuit you expect to find red, indicating high oxygen levels. I think everybody's pretty clear about where you find blue and red in this circuit, and where you find blue and red in this circuit, where you find blue and red in the heart chambers. I think everybody's pretty good about that. So then there's very little red. Wait, what does the red mean, everybody? The red means we have high oxygen concentration and a low concentration of CO2. What does the blue mean, everybody? It means that we have a high CO2 concentration and a low concentration of oxygen. Right? Everybody good? You already know where to find high oxygen and low oxygen. You already know where to find high CO2 and low CO2. And we're talking about the high and low, we're talking about the concentration of those gases, right? Okay. Then as long as you understand that, there's actually nothing new but a phrase that we have to introduce, this phrase. Otherwise I think you're good. Check this out. I've been telling you all along that fluids, that includes gases and liquids, are both fluid. They both flow. They're both fluids. Fluids are not just liquids. Fluids are liquids and gases. Anyway, all fluids exert an outward force on the walls of their container. Have we said that a lot of times before? If we're going to refer to that in its most generic form, we would call that outward force hydrostatic pressure. When it's blood pushing outwardly on the blood vessels, we call the hydrostatic pressure of the blood, we call it blood pressure. Everybody good with all that? That stuff I think we've said before. The hydrostatic pressure of the blood, we call blood pressure, right? Pushing outwardly on the walls of your blood vessels. Okay, well, think about that outward pushing force that the blood is exerting. Part of that pressure that the blood is exerting is due to the compressed gases that are in the blood. Oxygen is in the blood, and it's responsible for part of your blood pressure. If you took the oxygen out, you'd have less blood pressure. See what I mean? CO2 is in the blood, and it's responsible for a portion of your blood pressure too. It's a gas, a compressed gas in there. We know how compressed gases are. They're pushing outwardly on the walls of their container, and CO2 is in the blood. It's contributing partially to your blood pressure also. Wouldn't you kind of get that without me having told you that? You can already get that on 50. Well, the part of your blood pressure that oxygen is responsible for is called the partial pressure of oxygen. The partial pressure of oxygen is the part of your blood pressure that oxygen is causing. And we refer to it this way. P with a subscript O2. That's the partial pressure of oxygen. So let me ask you this. Would you expect the partial pressure of oxygen, the amount of the blood pressure that oxygen is responsible for, would you expect that to go up or down if the concentration of oxygen goes up? If there's more oxygen in your blood, do you think it's going to contribute more to the blood pressure or less? More. If there's more oxygen in the blood, it's going to be responsible for a greater part of your blood pressure. More oxygen in the blood, more pressure caused by oxygen. That's all. Everybody get it? In other words, the partial pressure of oxygen correlates directly with its concentration. The more concentration of oxygen in there is, the more it's going to exert pressure. That's part of your blood pressure. They correlate so directly that I think you don't even need to learn anything new. Because if you were ever asked this, hey, where on this picture would you find a high partial pressure of oxygen? If you ever asked that, all you have to do in your mind is substitute the phrase in the question. When it says partial pressure, you just have to substitute in your mind for concentration of it. It's the same question. Because they're the same. They correlate so directly. And if I asked you, where is there a high partial pressure of oxygen? Where in the bloodstream? Then you would think to yourself, he means, where is there a high concentration of oxygen? Where the red is. Everybody get it? Where is there a low partial pressure of oxygen? Well, where the oxygen concentration would be low. So if there's a high oxygen concentration, it's got a high partial pressure. If there's a low oxygen concentration, then there is a low partial pressure of oxygen. Nothing to it. But we also have to talk about the partial pressure of CO2. Which is written like this. The big P and the subscript CO2. Where do you think there's a high partial pressure of CO2 on the picture, everybody? In the blue or the red? In the blue. There's a lot of CO2, so CO2 is contributing more to your blood pressure in that place. Its partial pressure is higher because its concentration is higher. Where would we expect to find a low partial pressure of CO2? Well, wherever you see red, because there's not much CO2 in the blood there contributing, not much of it to contribute to your blood pressure. There's always some. Well, if you're okay with the idea that really all you ever have to do to understand this is just substitute in your mind concentration of for partial pressure of, then you would answer all these questions right. I could have asked you anything about whether there'd be high or low concentration of these gases before, and you would have known where you find them. Kelly, go ahead.
Does the pressure kind of stay relatively the same amount of pressure anyway? Because if there's more oxygen, there's less CO2 and vice versa.
Yes, so the pressure, the overall blood pressure isn't varying very much, but which gases are responsible for it is. Is that what you're asking?
Yeah.
Yes, absolutely. They try to show us this with these graphs. The trouble is that if I point to the graphs, you're going to think I want you to study the graphs. And I don't think you have to, in order for us to meet all of our objectives on a checklist in my mind called the Master Course Outline. All we really needed to do was provide a definition for partial pressure. I'm just trying to convince you it's so easy. Partial pressure. It's just a new phrase, partial pressure. It's the part of your blood pressure that those gases are responsible for. But the way that you're ever asked questions about partial pressure is you'd be asked, like, where in the circulatory system is it high or low for that particular gas? There's nothing new to learn there as far as that's concerned. So now, we have covered our one picture on the circulation story. And I think you'll already, because you already understand the blue and the red there, all we have to do is introduce the partial pressure. The last picture now is picture 25 in the chapter. And no, we're not going to get to it, except I want you to study it on your own. Don't I always do this? Except I want you to study it on your own, except let me give you a little bit of help. On figure 25, they're showing us internal and external respiration. Steps 2 and 4 of our 4-step process of respiration. External and internal respiration. Everybody see what I'm saying? See, external respiration and internal respiration, they're covered on figure 25. But they're covered in sort of like separate parts of the picture that I think are not adequately labeled. Look at part A of the picture and see there's a big red blood cell shown. It's in a capillary. And it's dropping off its oxygen out to your tissue cells while it's also picking up CO2 from your tissue cells. This exchange of gases between your blood and your tissue cells is this exchange of gases. We're talking about the exchange between the blood and the tissue cells, and we define that as being internal respiration. So part A of figure 25 should be labeled internal respiration. It's their term, internal respiration is. They just didn't use it in the most important place for it to appear in the book. They didn't put it there. Part A of figure 25 is called internal respiration. And then in part B of the picture, they're showing us this exchange of gases here between the blood and the alveolar airspace. So if we go back to 25, see in part B of figure 25, they're showing us a red blood cell in a capillary in the pulmonary circuit. where oxygen is coming in from the alveolar airspace, the alveolus, oxygen diffusing into the blood, CO2 is being dropped off out to the alveolar airspace. Remember this membrane that we make this exchange across? We call this the respiratory membrane. Alveolar epithelium, capillary endothelium, the fused basement membranes in between. This is external respiration. They just didn't label it as such, despite my email requests for them to do so. So when you're trying to study these on your own, please realize that A is talking about did I just say it wrong? A is talking about internal respiration, and B is talking about external respiration. This is external. Okay, please study figure 25 also. So next week then, we'll have a quiz on respiratory physiology stuff. I'm thinking like, figure like 10 forward would all be fair game. Because 10 was kind of new, including on tonight's quiz. So from figure 10 forward, oh, it's not that much. And then we'll cover this one more picture next week, and then we're going to cover the lymphatics.